Hi everyone, welcome back to the next video. We'll continue our discussion of evolution of this E. coli line in LTE to utilize citrate for carbon source, to utilize citrate as a carbon source for growth and energy. So, with the question before us, the conundrum in this evolution experiment is that let's say we have line 1 which has evolved for 80,000 generations, or if we talk of the time at which this happened, let's say this has evolved for 35,000 generations and this has no citrate utilization capacity. On the other hand, I have 11 such lines. So 11 such lines. On the other hand, I have this one line in which something seems to have happened such that the individuals in this population are able to utilize citrate. And the question is that why is it? And this question is more pertinent now because we know that another 50,000 generations have been processed and none of these 11 lines are still able to utilize citrate. So the question is that what happened in this line in terms of the genome of the individual which allowed it to utilize citrate? And there were two hypotheses of why it could have happened. In the first hypothesis, it was an extremely rare mutation that happened in this particular line which allowed for utilization of citrate. By rare mutation, what we mean is that let's imagine that this is the DNA of the organism, and maybe what needed to happen was that this part of the DNA needed to duplicate itself. Now, duplications happen all the time, but what made this rare was that this exact part of DNA needed to be duplicated. If it was slightly shorter, it wouldn't work, and if it was slightly longer, it wouldn't work also. So the exact same region of DNA needed to duplicate itself, and that is a very rare mutation. So maybe that's what happened, and maybe that happened somewhere around here in this line, and that's why we detected once that mutation happened, you had one individual in which it happened, and that individual had competitive advantage over the others in the population, and then it spread in the population, and that's why we saw that's why one morning at the time of subculture we saw that this flask was much more densely populated as compared to the other 11. That's one hypothesis. The second hypothesis is that there were a series of mutations leading up to a final mutation. None of these was rare. These are regular single nucleotide mutations. These set of mutations happen in a series in this particular line, leading up to a final mutation which conferred the ability to utilize citrate. So maybe these mutations had happened all along the way on this on this particular line, and finally a mutation happened, and when all 5 mutations were present together in a genome, the ability to citrate evolved. Only this mutation would not confer the ability to utilize citrate. It was only that this mutation, when in conjunction with these 4 previous mutations were present in the same individual, would that individual have the ability to utilize citrate. So, if these are the two competing hypotheses, then we need to think about that what is the experiment that one would do to test which of these two scenarios actually played out in the case of citrate utilization in LTE. To test this possibility, what was done in this study was that, remember, each of these lines we have freezer stocks made at every 500 generations or so. So, we have access to these frozen records of this evolution experiment that we can go back to, revive from the freezer stock and grow up again, and so on and so forth. So, the strategy that was used was that for each one of these 12 lines, if we go back a few thousand generations, let us say I go to 28,000. At 28,000 generations, I will revive this population in each one of these 11 lines and check for ability to grow in citrate. Check if grows on citrate. And obviously, it was found that there was no growth on citrate. That is not surprising, because even at 35 there was no growth on citrate. But when we do the same exercise for the line which actually did evolve the ability to citrate, we check if grows on citrate. And we found that there was no growth on citrate. Now, what that means is that at 20,000 generations there was no ability to grow on citrate, but at 35,000 for sure I know that this line is growing on citrate, these 11 lines are not. That means something happened between 28,000 and 35,000. Now, that is something that enabled utilization of citrate in this particular line could be either this hypothesis or this hypothesis. So, what would be the experiment here? The experiment that was done to check which of these two hypotheses played out was that now if I go back to the freezer stock from 28,000 and evolve again all these 11 lines and this particular line. Okay. So, remember, this is happening like this, that there is an evolution experiment going on. This was 28,000 generations. At this point, I made a sample in a vial and I stored it in minus 80 degrees Celsius freezer. While the experiment continued via this transfer every day, this continued, and I am at 35,000 now. So, I have the culture that was growing on this day stored away in minus 80 degrees Celsius that I can go back to, revive, and then start a parallel evolution experiment again from this point only. So, let us do this on a fresh slide. This is, let us say, 35K, this is this is 28K, this is 11 lines, and the line where the ability to utilize citrate did evolve, let us say, is this is 35K, this is 28K, and so on. This is one line. So, if hypothesis 1 is right, if hypothesis 1 is correct, then ability to utilize citrate came as a result of a very rare mutation, a very rare single mutation. If that is the case, then this rare mutation could have happened in any line. Could have happened in any of the 12 lines, because it's so rare, and it only happened in this particular one because this was the fortuitous one which happened to acquire that mutation. But really it could have happened in any one of, there was no particular reason that it needed to happen in this particular line. So the experiment to test this is that if I take the 28,000 stock from these 11 lines and evolve them again, and I take the 28,000 stock which I know is not growing on citrate. So by 28,000 that rare fortuitous mutation had not happened. If I take this and I evolve this again, then the question I ask is that which line should acquire the ability to utilize citrate. And if hypothesis 1 is correct, then the ability to utilize citrate is conferred because of this very fortuitous single rare mutation, and that single rare mutation could happen in any one of those 12 lines. So the ability to utilize citrate could come up or let us say should come up in any one of the 12 lines, does not need to be one, in any of the 12 lines, should hypothesis 1 be correct, because that rare mutation can happen anywhere in any line, in any individual, in any of these flasks. So there is no reason that it should again arise in the line which had already acquired the ability to utilize citrate. So every line has the same probability of evolving to utilize citrate. So that is the experiment that they did to check whether hypothesis 1 was true or not, and if hypothesis 1 was true, this is what is our null expectation. Suppose the outcome from this was that when we redid the evolution experiments, line 3 and line 5 and line 9 evolved the ability to utilize citrate, whereas in the first case it was line 7. Then hypothesis 1 would bear out that this rare mutation is now happening at some other place. Of course, it is rare. We are saying that this has to be a very rare and a lucky mutation. So this evolution has to be carried out for a long time for us to statistically be able to confidently say that this is what is happening. It turns out that literally trillions of cells were checked for their ability to utilize citrate, and in these 11 lines the number of cells that evolved the ability to utilize citrate was 0. So not even once did these 11 lines acquire the ability to utilize citrate even when started from 28,000 generations. On the other hand, when individuals from this particular line were started for this re-evolution experiment from 28,000 generations, 19 different times individuals were found which acquired the ability to utilize citrate as this repeat evolution experiment was done. So this gives us a pretty clear indication that it is not hypothesis 1, it is in fact hypothesis 2 which is responsible for the ability to utilize citrate in this one line. Because what is happening is that this line already has these mutations available, and all it needs now for it to utilize citrate and exhibit higher growth rates is this one more mutation to happen, and sooner or later one more mutation will happen, and that shows as increased density in the culture media that we are using. On the other hand, these mutations can happen here, but they will have no effect because these other prior mutations that are needed for this functionality have not happened in these 11 lines. So, through this it was found out that ability to utilize citrate was actually acquired through a series of mutations and not just one very lucky rare mutation in this particular flask. And then it took a few years, but a few years later we found out the exact mechanism as to what had happened, which led to this flask exhibiting growth on citrate. And it's actually, we won't go into much detail, but broadly what had happened was the following, that the gene which encodes for transport, which encodes for the protein which is responsible for transport of citrate is called CYP. Now, CYP, its gene expression, first transcription and then translation and then getting embedded in the membrane, and then bringing citrate in. This is controlled by the region upstream, which is called the promoter region of CYP. And this promoter region does not allow for transcription in oxic conditions. So, this promoter region only allows transcription in anaerobic conditions. As a result, the cells of E. coli in LTEE, they do not make this protein and they do not have the CYP transporter embedded in the membrane, which is bringing in citrate. So, what happened in the line that eventually exhibited the ability to utilize citrate was that this gene was actually duplicated into 2. So, now this evolved strain has 2 copies of CYP, and via this duplication event, in one copy its expression was driven by the CYP promoter, which is the same promoter as here, but in the duplication event only this part was duplicated, and upstream of this gene was another promoter which was not CYP's promoter, but this is another promoter that was present here on the DNA. So, this is another promoter. And now, even in the culture conditions that we are talking about, growth is happening on citrate because this promoter is not dependent on anaerobic conditions to facilitate transcription. This promoter is actively transcribing, and then the transcript is being translated and we get CYP protein, and that leads to metabolism of citrate as a carbon source and growth to take place. So, it turns out that even this much, even this fortuitous mutation of duplication taking place, but if duplication had taken place starting from this point, then its own promoter would be copied again, and that would be driving transcription, and that would not work because this promoter does not allow transcription in oxic conditions. So, this was the fortuitous event that needed to happen, but it turns out that even with this genome, the growth rate benefit that is conferred is only about 1%, and that is that is barely anything. That is a very small mutation. Remember that tells me that this is S equal to 0.01. This mutation, when it happens, only has 1% chance of surviving drift, as we have discussed in the class, that probability of surviving drift is actually just equal to S. So, this mutation per se does not confer a huge benefit to the individual. However, what happens is that more mutations take place after this, which lead to further increase in fitness. So, this is the molecular details of what happened in this case, which allowed one of these lines to exhibit growth on citrate as carbon source. So, now these complex evolutionary processes are thought to happen via 3 different steps. The first step is called potentiation. Potentiation is, again, let us go back to the evolution experiment, and this is at 35,000 generations. Potentiation is the occurrence of these prior mutations which did not confer the ability to utilize citrate, but it brought the cell to such a point such that acquisition of one more precise mutation and the cell would become Cit plus, which is how we refer to citrate utilizing cells. Up until this mutation, the cell was Cit minus. But what these potentiating mutations did was that it brought the cell to a point that one more and it would become Cit plus. If these had not happened, then this mutation would not be able to make the cell Cit plus. So, these are called potentiation mutations. The second step then is actualization. So, these 3 collectively belong to this group. The second is called actualization, which is the mutation that actually changes the phenotype from Cit minus to Cit plus. But actualization mutations will only realize this transition if they happen after the potentiation mutations have taken place in the genome of the organism. And third set is called refinement. And in refinement, what we saw that even with that duplication event in Cit-T, this was only growing 1% better as compared to the Cit minus cells. So, the advantage is not huge, but something novel has happened. The novelty is still bad and it can be improved upon by subsequent mutations. So, these are the refinement mutations that happen after actualization has taken place, which further improve the trait that has been evolved as a result of potentiation and actualization mutations. So, these are roughly the 3 steps in which these complex traits are thought to evolve. One thing that this experiment should make abundantly clear is that so far we have been saying that evolutionary fate is decided by selection what sort of environment did this population grow on is an extremely important, but also drift or chance events. That is what we have been discussing so far. However, what this experiment also shows us that there is an extremely important determinant of evolutionary processes, which is the history of the population, the evolutionary history of the populations. And this was abundantly clear in the citrate example, because we saw that you had a line, ability to utilize citrate was only possible because in this particular line these potentiating mutations had already happened in its evolutionary history. On the other hand, in these other lines, other mutations had happened which were not potentiating with respect to citrate utilization. So, at this point, when I do this re-evolution experiment from the freezer stock, even though, so at this point this is sit minus, at this, these are the 11 lines, this is the one line, at let us say this is 35,000 generations, this is sit plus and this is sit minus. However, if I go back in time and go back to 28,000, then even this is sit minus and this is also sit minus. But when I re-evolved them, one of them acquired the ability to transition from sit minus to sit plus 19 times, whereas the other 11 combined transitioned from sit minus to sit plus 0 times. And that reason is because of the evolutionary history of these populations, of what has been their trajectory which has brought them to this particular point in these 30,000 generations or so. So, evolutionary history is extremely important in the context of what can and cannot be achieved via evolutionary change by populations. In fact, in the early days of LTEE, there is a classical paper in Science in 1995 by somebody called Travisano and Lenski, which talks about very simple basic experiments which allow us to figure out that how much of evolution is done by selection, how much of evolutionary change is brought about by chance, and how much of evolutionary change is brought about by the evolutionary history of the population. We will discuss this particular paper in our next video. It is also a paper which is great illustration of how, by asking very precise questions and simple experiments, we can find insights which are very deep into evolutionary questions. We will continue with that discussion in the next video.